Howard, I have to go to the bathroom and no one will take me home.
What's wrong with the bathroom here?
Pneumococcus, streptococcus, staphylococcus, and other assorted caucuses.
Sheldon, my mother is on her deathbed and my fiance is grief-stricken over putting...
Hi and welcome. I am Utsab. I am a grad student here at the University of Georgia, and we study Microbiome Bioinformatics, and we do a lot of predictive modeling based on those microbiome datasets. So this is a video series about Microbiome Bioinformatics with QIIME. So we will be using QIIME as a tool to create insight from your microbiome data, starting from the FASTQ files. So welcome. So without further ado, let's dive in. So what exactly is microbiome? So in simple terms, a microbiome is a collection of microorganisms and their genes living in a particular environment. So these environment can be soil in your backyard to the oceans, from panda's gut to yours. So you are never truly alone. You carry trillions of microbial friends wherever you go. Let's break down a few examples of microbiomes to paint the picture. Let's say human microbiome. We humans host diverse microbiomes on our skin, in our mouth, up to nose, especially in our gut. So the gut microbiome, the community of microbes in our digestive tract, is one of the most studied. It's like bustling metropolis of bacteria. These microbes helps us digest food, train our immune system, and even produce vitamins. Another example is environmental microbiomes. Microbes basically cover the earth. Soil microbiomes in the forest help cycle the nutrients for plants. Ocean microbiome, like phytoplanktons and bacteria, produce a huge portion of oxygen that we breathe in. Even in extreme places like hot springs or deep sea vents, they have unique microbial communities. If there is a habitable niche, microbes have likely colonized it. So why do these microbiomes matter? Because microbes, despite their tiny size, have big impacts on the environment and the host. Let's take example of humans. In humans, research has linked gut microbiome to wide range of health conditions, from obvious ones like digestive diseases to surprising ones like the connections with the obesity, diabetes, immune disorder, mood, even how cancer patients respond to treatments based on the microbiomes. In short, microbiomes are everywhere and affect everything. Appreciating their importance sets the stage for why do we want to study them. And that's where the 16S rRNA gene comes in, our secret weapon for exploring this microbial world. So the 16S rRNA gene, it's like a barcode for bacteria. You scan it and you know which bacteria is that one. So in short, it is called 16S only, instead of calling it 16S rRNA gene. This gene is the rock star of microbiome research for bacteria and archaea. But what makes this single gene so special? Why do we use 16S to identify microbes? Think of 16S gene as a barcode or fingerprint that nature has provided bacterial DNA. Just like every product in the grocery store has a unique barcode, every species of bacteria has a unique 16S sequence signature. So, well, mostly unique and close enough to tell a different group apart. So talking about the 16S rRNA gene, there's a paper published in 2022. This is the paper by Dr. Lorenzo. So in the paper, there is a schematic representation of 16S rRNA gene, which looks like this. There is a DOI to this paper if you want to read this paper. This is one of the best paper if you want to get started with metagenomics or 16S rRNA gene sequencing. So let's say this is a 16S gene. So it will have two types of regions. The first one is variable region. The second one is conserved. So in 16S gene there are 9 variable regions. It is named as V1, V2, the last one is V9. Okay, and the region in between those variable regions, this region, let me change the color. This region, these are conserved regions. So if we look at bacteria 1, bacteria 2, bacteria 3, let's say these are different bacteria. If we look at the first conserved region, the nucleotide base, the sequence, this is just an example. It can be A, T, G, C, C, A, let's say conserved 1, T, G, C, C, A, T, G, C, C, A. I'm just trying to make things easier to understand. These segments are longer than just 1, 2, 3, 4, 5, 6 nucleotide. So the conserved region is universal. We can use these conserved regions to have a broad range of primers. That can target bacteria and archaea. And if we look at the variable region, this or this or any of the variable region, then let's say in the variable region, this is bacteria 1, bacteria 2, bacteria 3. So let's assume we are using V1. It will be AT, GC, CA, something like this. TA, GG, A, something like this. AGG, GAT, something like this. So the variable region will differ in between the bacteria, unlike the conserved region. So they are like fingerprint. Each bacteria will have a unique nucleotide sequence in the variable region. So due to the presence of conserved regions, we can have a broad range of primers, and due to the variable regions or the hypervariable regions, we can use them as marker for identification. For example, this bacteria can be E. coli, this can be pneumococcus or something like that. So this is the reason why 16S gene is called marker gene. We can use the variable regions to identify different bacteria, different microorganisms. Now that we know why we focus on 16S, let's zoom out and see how a typical 16S microbiome study is done. So what happens between obtaining a sample and getting your data analyzed? So I'm going to give a high-level overview of the microbiome workflow. So the first is sample collection. Everything starts with collecting your samples. So this could be stool samples for your gut microbiome, soil scoop for environmental microbiome, swabs from skin, water from a lake, and anything that contain microbiome. So a good sample collection means keeping things sterile, avoiding contamination, and often freezing and preserving the samples quickly to keep the DNA intact. So the next step, the second step, is DNA extraction. So next you have got to get the microbial DNA out of the sample. This often includes physically breaking apart the cells and enzymatically breaking apart the cells. The third step is PCR amplification of 16S rRNA gene. Now we use the PCR to make many copies of the 16S gene regions we care about. We add primers, which is short DNA sequences that matches the conserved region flanking a variable region. Through PCR, those 16S segments from every bacterium in the sample get copied millions of times. We also typically add index or barcode sequences at this step, not to be confused with the 16S being a barcode of species. Here the barcode or index means a short DNA tag that is used to label which sample is which. I mean, sample 1 is from this animal or this human, sample 2 is from this human, something like that. We just index the sample so we can identify after the sequencing. So this indexing or barcoding allows us to mix many samples together in one sequencing run, but still tell the reads apart by sample letter. After PCR amplification of 16S, the next step is sequencing. So the pooled indexed PCR products, which are called amplicons now, are then put on a DNA sequencer, often Illumina machines. Think of the sequencer as a DNA reader that splits out a big file of the sequence data, which is the letters A, T, G, C. So for paired end sequencing, we'll get two FASTQ files per sample, one for each read, one for forward, one for reverse. And for single end sequencing, we will only get one file per sample. The next step after sequencing is bioinformatics analysis. This is where QIIME and similar tools come in. We will be using QIIME to process the raw sequence files into valuable insights. So the next step after bioinformatics analysis is biological interpretation. After all the data is crunched, you will interpret the results in the context of biology. Are certain microbes more abundant in one group versus the another ones? Like if certain microbes are present in higher abundance in the healthy individuals compared to the diseased ones. So ultimately you tie it back to the original questions or hypothesis that motivated the study, right? Don't worry if not everything makes perfect sense yet. We will dive deeper step by step in later videos. The key takeaway is that 16S microbiome study involves both wet lab as well as dry lab. So we need to process the sample, do the sequencing, and then crunch the data and create biological insights from that data sets. So if the terms are new to you, so you don't have to worry about it. Throughout the series, each step will be demonstrated with actual commands and the output, and you will be provided with the data and the commands. You just have to execute and see the outputs. So by the end, you will have taken raw 16S sequence data, which I will provide you through all the way to the biological insight. So in the next video, we will get practical, setting up QIIME on your computer, meaning installing QIIME 2 on your computer. I will guide you step by step, but I assume that you have basic command knowledge on Linux. I mean, you can use simple commands like cd, ls, mkdir in the Linux system or terminal. So if you do not have any experience with basic Linux commands, then I'm attaching a document with the most used Linux commands and what they do. Just go through it before watching the next video. It's just a page. So thank you for watching. See you in the next video.